Hey Mustangs, in this video we're going to take a look at meiosis. We're going to see how we create these types of cells here. So here is a gamete, and this big old thing is a gamete here. These little ones are of course sperm, and the large one that they're trying to get into is an egg. So how do we create sperm and egg? Meiosis is the process that helps us do that. Alright, first a quick introductory video about the discovery of meiosis.
Was studying the cell division process, biologists already knew that fertilization resulted from the union of two sex cells, sperm and egg. What they didn't know was why these were the only types of cells in the body capable of reproducing new life. What made them different? The first answer was provided in 1883 by Belgian zoologist Edward van Beneden. Van Beneden was studying a species of roundworm when he discovered that nearly all of its cells contained 4 chromosomes. The only exceptions were the roundworm's sex cells, the male sperm and the female egg. These cells had only 2 chromosomes. Van Beneden observed that once 2 of the sex cells came together, a full set of 4 chromosomes appeared in the fertilized egg. But just why the sex cells had only half the number of chromosomes compared to all the other cells was still a mystery. Then in 1887, German biologist August Weismann entered the picture.
He knew about the work of Van Beneden, who had shown that the sperm and the egg bring in equal numbers of chromosomes. But Van Beneden's work had not really worked out the details of how the chromosomes were reduced in number.
He saw them go in half, and he saw them leave them on.
He saw that the sperm and the egg brought in half the number of chromosomes as the body cell, but he didn't really know how they got rid of the half that they got rid of. So Weismann discussed how this might happen from a theoretical standpoint. And he said, well, what you could have would be that either every one of the chromosomes would just split in half, and half would go off one way and half would go another way, or you could have it more complicated, that the chromosomes could pair, and then instead of splitting, they just separate again.
After years of studying the behavior of chromosomes, Weismann determined that at some point a developing organism signals its sex cells to divide their number of chromosomes in half. Weismann called this very special form of cell division meiosis.
I think they will look back and see this as the golden age of biology, because, let's say, 200 years, between 1800 and 2000, we went from not knowing what distinguished a stone from a frog, okay? And now we know the difference between a stone and a frog.
That's pretty good.
So that, in a real sense, we've answered the old philosophical question, what is life? Maybe not to everybody's satisfaction, but we're well on the way.
Alright, so let's take a look at what August Weismann figured out. So, 3 questions I want you to think about as you watch this video. What's the point of meiosis? What's made at the end of meiosis? Are the cells at the end of meiosis haploid or diploid? So, if we take a look, we know how a diploid cell is made. Our original diploid cell, the zygote, how that's made. A haploid egg and a haploid sperm come together. And in the egg, for every chromosome that's in the egg, there's a matching chromosome in the sperm. So you can actually see this when you look at this sperm and this egg here. So when they join together, they create a diploid cell, a cell that has homologous pairs. That's what a diploid cell has. Remember, di for 2 in a pair. So diploids have homologous pairs. So we know that if we join 2 haploids together that have matching chromosomes, you will form a diploid cell. So what we want to do is we want to go in reversed. How do you go from a diploid cell to haploid cells? In this case, at the end of meiosis, we go from a diploid cell, that's what we start with, an actual diploid cell, and we wind up making 4 haploid cells. So how do you go from here to there? All right, so for meiosis, we have our starting cell. There's an interphase for meiosis. There's prophase 1, metaphase 1, and if you remember PMAT, you should be able to finish the rest of this on your own here, but of course I will help you out. Anaphase 1 and telophase 1. But meiosis doesn't stop there. It has another series of 4 phases. So we have prophase 2, metaphase 2, anaphase 2, and telophase 2. So again, looking at the sperm and the egg, this is what we want to create. We want to create haploid cells at the end of meiosis. We do not want any pairs in the egg and no homologous pairs in the sperm. Yet we're starting with a diploid cell that has homologous pairs. So how do we get haploid out of that? Now, if we did mitosis, that would not work. Because with mitosis, you start with a diploid cell and you end with 2 diploid cells. We don't want that. So mitosis isn't going to work to create sperm or egg. So the first 4 phases of meiosis, they are designed to make sure that we separate our homologous pairs. Because again, we don't want our sperm or egg to have homologous pairs. We want them to be haploid, 1 from each pair. So these first 4 phases here separate homologous pairs. So here we have 2 homologous pairs. So this pair, during these first 4 phases, we're going to separate these 2 from each other and put them into different cells. Same thing here, we're going to put these 2 into different cells. So these first 4 phases, prophase 1 through telophase 1, are all about separating homologous pairs. The next 4 phases, these 4 phases are to separate sister chromatids from each other. Because right here we see there's an interphase. So that means the cell's going to grow bigger, make a copy of every chromosome before it enters into meiosis. So that means it's going to have sister chromatids. It's going to have copies of chromosomes. So the last 4 phases are all about separating those sister chromatids from each other. Okay, first 4 separate homologous pairs. The second 4 phases are about separating copies, sister chromatids from each other. Alright, so interphase, this is when we make a copy. So if you remember what our starting cell looked like, here's what that starting cell will look like after we make a copy of each chromosome. So during interphase, the cell grows bigger and a copy of each chromosome is made. So the copies, sister chromatid, are still connected together at the centromere. Now this star, the reason I put a star on this represents the chromosome from dad. This is the one from mom. This is the chromosome from dad. This is the one from mom. So we're going to track them as we move through meiosis. So the first phase of meiosis is prophase 1. So during prophase 1, chromosomes condense and a process called crossing over occurs. Metaphase 1, well, remember, metaphase means middle, so during metaphase 1, the chromosomes line up in the middle, but different from mitosis. In mitosis, they were in one long straight line. But remember, the first 4 phases are all about separating homologous pairs. So homologous pairs line up in the middle. Oops, my bad. Homologous pairs line up in the middle, side by side. So this is a homologous pair, and this is the homologous pair. They line up side by side. They're still lined up in the middle though, side by side. Next, anaphase, remember A away, okay? But in this case, instead of pulling sister chromatids apart, anaphase 1 pulls homologous pairs apart. So it pulls this chromosome from this homologous pair to this side, pulls this chromosome of that homologous pair to this side. So we separate the homologous pairs during anaphase 1. During telophase 1, coupled with cytokinesis here, the cell has split in 2 and chromosomes kind of unwind a little bit. Then we hit the second 4 phases. So we have prophase 2. During prophase 2, well, not much happens. Chromosomes start to recoil again, get condensed, and new spindle fibers begin to form. Metaphase, M means middle. So right here, the chromosomes line up in the middle of the cell. Anaphase 2, remember AOA. So we have sister chromatids now getting pulled apart. Copies that are being pulled apart from each other. One goes one way, one goes the other way. And you can imagine what's going to happen next. It's going to pinch right down the middle here. And this will form one cell, this will form another, this will form one cell, that will form another. So sister chromatids are pulled apart during anaphase 2. And then finally you wind up with telophase 2 here coupled with cytokinesis. You wind up with 4 identical, unidentical, whoa, big idea there. We wind up with 4 unidentical haploid, in this case sperm cells. Okay, and you can see that they're unidentical. These over here are not the same as these over here. Okay, you can see this one's from dad, that's from mom, that's from mom, this is from dad, that's from mom. So the DNA has been kind of mixed up a little bit here. Right, so each cell separates into 4, leaving you with unidentical haploid sex cells or gametes. Alright, so you should be able to answer these questions now. What is the point of meiosis? What is made at the end of meiosis? Are the cells at the end of meiosis haploid or diploid? And a quick overview of meiosis as a visual aid here.
The process of meiosis results in the formation of 4 daughter cells, each containing half the number of chromosomes found in the parent cell. In this case, the parent cell has 4 chromosomes, and the daughter cells have 2 chromosomes. The chromosomes are shown here as they look during cell division. In the parent cell, the chromosomes have replicated and condensed. Each chromosome consists of 2 sister chromatids joined at the centromere. The parent cell has 2 sets of chromosomes. One set of chromosomes, colored red here, came from the mother. The other set, colored in blue, came from the father. The matching pairs of chromosomes, one from each parent, are called homologous chromosomes. Homologous chromosomes carry genes for the same traits and are the same size and shape. A cell that contains 2 complete sets of chromosomes is called a diploid cell. The parent cell divides twice during meiosis. The first cell division is called meiosis 1. During meiosis 1, the homologous chromosomes are separate. 2 new cells are formed, each containing half the chromosomes from the parent cell. A cell with only 1 set of chromosomes is called a haploid cell. The second cell division is called meiosis 2. During meiosis 2, the sister chromatids of each chromosome separate. The 4 daughter cells are also haploid cells.
Alright, so the point of meiosis is to create sperm and egg. So you start with the diploid cell. You first separate homologous pairs, and then you separate sister chromatids, and you wind up with 4 unidentical haploid sex cells, either sperm or egg.